A malaria vaccine is a vaccine that is used to prevent malaria. The only approved vaccine as of 2015 is RTS-S, known by the brand name Mosquirix. It requires 4 injections and has a relatively low efficacy. Due to this low efficacy, the World Health Organization does not recommend the routine use of the RTS-S vaccine in babies between 6 and 12 weeks of age. A WHO-led implementation program is piloting the vaccine in 3 high malaria countries in Africa. In 2019, the first phase of the project, covered by grants from UNITAID, Gavi and the Global Fund, is planned to establish the feasibility, impact and safety of RTS-S when used as part of a routine immunization program. Research continues into recombinant protein, attenuated whole organism and viral vector vaccines. RTS-S, with support from the Bill and Melinda Gates Foundation, is the most recently developed recombinant vaccine. It consists of the P. falciparum circumsporozoite protein from the pre-erythrocytic stage. The CSP antigen causes the production of antibodies capable of preventing the invasion of hepatocytes and additionally elicits a cellular response enabling the destruction of infected hepatocytes. The CSP vaccine presented problems in trials due to its poor immunogenicity. RTS-S attempted to avoid these by fusing the protein with a surface antigen from hepatitis B, hence creating a more potent and immunogenic vaccine. When tested in trials, an emulsion of oil in water and the added adjuvants of monophosphoral AN/QS21, the vaccine gave protective immunity to 7 out of 8 volunteers when challenged with P. falciparum. RTS-S-AS01 was engineered using genes from the outer protein of P. falciparum malaria parasite and a portion of a hepatitis B virus plus a chemical adjuvant to boost the immune response. Infection is prevented by inducing high antibody titers that block the parasite from infecting the liver. In November 2012, a phase 3 trial of RTS-S found that it provided modest protection against both clinical and severe malaria in young infants. As of October 2013, preliminary results of a phase 3 clinical trial indicated that RTS-S-AS01 reduced the number of cases among young children by almost 50% and among infants by around 25%. The study ended in 2014. The effects of a booster dose were positive, even though overall efficacy seemed to wane with time. After 4 years, reductions were 36% for children who received 3 shots and a booster dose. Missing the booster dose reduced the efficacy against severe malaria to a negligible effect. The vaccine was shown to be less effective for infants. 3 doses of vaccine plus a booster reduced the risk of clinical episodes by 26% over 3 years, but offered no significant protection against severe malaria. In a bid to accommodate a larger group and guarantee a sustained availability for the general public, GSK applied for a marketing license with the European Medicines Agency in July 2014. GSK treated the project as a non-profit initiative, with most funding coming from the Gates Foundation, a major contributor to malaria eradication. On the 24th of July 2015, Mosquirix received a positive opinion from the EMA on the proposal of the vaccine to be used to vaccinate children aged 6 weeks to 17 months outside the European Union. Pilot project for vaccination has been launched on the 23rd of April 2019 in Malawi, on the 30th of April 2019 in Ghana, and on the 13th of September 2019 in Kenya. The task of developing a preventive vaccine for malaria is a complex process. There are a number of considerations to be made concerning what strategy a potential vaccine should adopt. P. falciparum has demonstrated the capability, through the development of multiple drug-resistant parasites, for evolutionary change. The Plasmodium species has a very high rate of replication, much higher than that actually needed to ensure transmission in the parasite's life cycle. This enables pharmaceutical treatments that are effective at reducing the reproduction rate, but not halting it, to exert a high selection pressure, thus favoring the development of resistance. The process of evolutionary change is one of the key considerations necessary when considering potential vaccine candidates. The development of resistance could cause a significant reduction in efficacy of any potential vaccine, thus rendering useless a carefully developed and effective treatment. The parasite induces two main response types from the human immune system. These are anti-parasitic immunity and anti-toxic immunity. Taking this information into consideration, an ideal vaccine candidate would attempt to generate a more substantial cell-mediated and antibody response on parasite presentation. This would have the benefit of increasing the rate of parasite clearance, thus reducing the experienced symptoms and providing a level of consistent future immunity against the parasite. By their very nature, protozoa are more complex organisms than bacteria and viru.